Am I visible and audible to all of you? Sorry for the disturbance. थोड़ा सा शायद नेटवर्क इश्यू था. Am I audible now? So we have different type of lipid like phospholipid, we have glycolipid, and we have last lipid, that is our carbohydrate, sorry, cholesterol. It can be saturated or unsaturated fatty acids. We have different type of protein there, transmembrane protein, we have this lipid anchored protein, we have the peripheral proteins. Is this clear with everybody? So moving further to the carbohydrate, they are the minor component of our cell membrane. They are present 5% in our cell membrane and they provide membranal asymmetry and they are responsible for the immune reaction. So moving further, protein concentration varies with the different cell. Normally the protein is 55%. So normally the protein is 55% while lipid is 40%. Okay? But there are some membrane which are exception. There are some membrane that consist of high protein and lipid concentration, and in that we have this inner mitochondrial membrane that consists of 76% protein. Our presynaptic membrane consists of 70% protein. Next is the high lipid and protein concentration. There are some membrane जहाँ पे lipid concentration is more, like our Schwann cell membrane, our oligodendrocyte consists of more lipid concentration. 76% lipid is present in their cell membrane. So remember, our nerve cell membrane have high lipid to the protein concentration. तो नट सेल में रिवाइज कर लेते हैं. आई गेस वॉइस क्लियर नहीं था, तो नट सेल में हम पूरा रिवाइज कर लेते हैं सेल मेम्ब्रेन को, फिर फर्दर प्रोसीड करते हैं सो दैट यू ऑल कैन आंसर द सेशन. Fluid mosaic model is the most commonly accepted model for the cell membrane. In this, there is a lipid bilayer in which proteins are embedded. The major component is protein. 55% is protein, 40% is lipid, and carbohydrate is only and only 5%. When we are talking about the weight basis, lipid and protein are present into equal ratio. That is 1 is to 1. This is the diagram. This is the phospholipid layer, and different type of proteins are there: peripheral protein, integral protein, and the glycoprotein are there. Integral protein can be either the channel protein or the carrier protein. The phospholipid has the phosphate head and the fatty acid tail. The phosphate head is hydrophilic and fatty acid tail are hydrophobic. So this phospholipid is antipathic lipid. So lipid, now coming to the lipid in our cell membrane, the lipid can be either phospholipid, glycolipid, or the cholesterol. Different type of phospholipid are there in our membrane. First is phosphatidylcholine, which is a major lung surfactant. Next is the sphingomyelin, that is a minor lung surfactant. Next one is phosphatidylserine. This is present at inner cell membrane, and whenever it is present at the outer side, it is going to signal the apoptosis, which is a programmed cell death. Next is the phosphatidylinositol that act as a secondary messenger for the IP3 diet calcium. Next is the cardiolipin that is present in mitochondrial membrane of human heart, and whenever the anti-cardiolipin antibody is present, they are present in syphilis, and the VDRL test is done for this measurement. Coming to this, next one is the glycolipid. This glycolipid has two forms. We have ganglioside, we have cerebrocyte. Next is the cholesterol. The cholesterol is going to provide the membranal fluidity, and they act as a precursor for vitamin D. In this, we have two types of fatty acid, unsaturated and saturated. So unsaturated fatty acid are the essential fatty acid. They are only going to increase the membranal fluidity. For example, we have linoleic acid, linolenic acid, and arachidonic acid. Saturated fatty acid like trans fatty acid is going to decrease the membranal fluidity. In this, we have this stearic acid and the palmitic acids. Now coming to the protein in our membrane, 40% is the lipid, 55% is our protein. So protein may we have this transmembrane protein, we have peripheral protein, we have lipid anchor protein. Transmembrane protein is also known as integral protein. It can either be the channel or the carrier. So it can act like either the receptor like G protein coupled receptor, or it can act like a transporter like sodium potassium pump. Next is the peripheral protein. So peripheral protein are supportive in nature. They provide the structural integrity of cells. For example, RBC have this spectrine and carrying the peripheral protein. Skeletal muscle is having this dystrophin. So whenever the mutation occur in this peripheral protein, the structural integrity of cell will be hampered. And so that's why mutation in these protein like spectrine is leading to the hereditary leptocytosis, and chirin mutation is going to hamper the same. That is, structural integrity of RBC will be hampered in this situation, and it is going to lead this hereditary spherocytosis. Dystrophin mutation is going to lead the structural integrity hampered in skeletal muscle, and that is going to lead this duct and muscular dystrophy. So coming to the lipid anchored protein, this lipid anchored protein is also known as glycosyl phosphatidylinositol protein. In RBC, we have CD55 and CD59. They are the decay accelerating factor. They are present on the RBC membrane. So they prevent the complement attack. And that's why whenever this decay accelerating factor is going to be absent, or there is a defect in this decay accelerating factor, what happened? Complement attack on RBC will be there. Hemoglobin will be released in the urine, and that's why it leads to the paroxysmal nocturnal hemoglobinuria. So moving further, the last component is the carbohydrate. Carbohydrate is the minor component. Only 5% of carbohydrate is present in our lipid. They provide asymmetry to the membrane, and they are responsible for immune reaction. Next, this is also important. Normally the protein concentration in our cell membrane is 55%, lipid is 40%, but there are some exceptions. Some membrane consists of high protein and lipid ratio, more than 55% protein is there. So in this we have inner mitochondrial membrane and the presynaptic membrane. And there are some membrane where lipid and protein concentration ratio is more. And this we have the nerve cell membrane like Schwann cell membrane and the oligodendrocyte. Is this clear with everybody? So this is all about cell membrane. Anybody, any doubt? Do reply fast. Anybody, any doubt? Okay, moving further to the cellular organelles and the cellular junction. So first is the endoplasmic reticulum. we can have this rough endoplasmic reticulum, we can have this smooth endoplasmic reticulum. Two type of endoplasmic reticulum are there. They are known as synthetic machinery of cell. Protein synthesis, lipid synthesis, steroid synthesis all occurs in our endoplasmic reticulum. So that's why they are known as synthetic machinery of cell. So first coming to this rough endoplasmic reticulum. Why it is known as rough endoplasmic reticulum? Because of presence of ribosome, the surface is rough, and that's why they are also known as granular endoplasmic reticulum. So they are basically involved in synthesis of protein, and they are also involved in folding of protein. So this point is important. This folding of protein occur with the help of heat shock protein, which is also known as chaperones. ठीक है, फर्स्ट फंक्शन इज synthesis, second is the folding of protein, and the misfolded protein is also degraded in rough endoplasmic reticulum, and that phenomena is known as endoplasmic reticulum assisted degradation. So moving further, what happened? This ubiquitin molecule combined with this misfolded protein. Do you remember? This ubiquitin is important for degradation of misfolded protein. This ubiquitin is going to combine with the misfolded protein, and they will move to the proteasome, and the protein destruction occur out there. So whenever there is an absence of this endoplasmic reticulum assisted degradation, what happens? Misfolded protein will accumulate in our body, and that lead to in certain cases it lead to Brand's disease. In certain cases, it leads to A beta amyloid formation that is causing this Alzheimer's disease. So this misfolded protein should be degraded in this endoplasmic reticulum. So what is the role of endoplasmic reticulum? Protein synthesis occur, okay, folding of protein occur, and misfolded protein is also degraded by this endoplasmic reticulum. Coming to this next one, that is smooth endoplasmic reticulum, means out here ribosomes are not there, so that's why it is known as agranular endoplasmic reticulum, and they are involved in lipid and steroid synthesis, and they are responsible for storage and metabolism of calcium. They are also responsible for catabolism and detoxification of toxic substances. Next is the mitochondria. The mitochondria is also known as powerhouse of cell. Why they are known as powerhouse of cell? Because they are involved in synthesis of energy rich compound ATP. ठीक है, ATP synthesis के लिए responsible है. That's why it is known as powerhouse of cell. Now coming to its inheritance, it is inherited from the maternal side, and this mitochondria has its own genome, and this mitochondria is also responsible for apoptosis initiation as well as regulation of apoptosis is done by mitochondria. Is this clear? Moving to the next cellular organelle, that is the Golgi apparatus. This Golgi apparatus consists of three parts: cis phase, trans phase, and the Golgi apparatus proper. First is the cis phase. They receives the proteins, that's why it is known as the receiving end. To receive the protein, it is closest to the rough endoplasmic reticulum. Next is the trans phase. From there the our glycosylated protein is released, that's why it is known as the releasing end, and that is farthest from this rough endoplasmic reticulum. Next one is Golgi apparatus proper, that is also known as functional end, because actual Golgi apparatus function is done out here. What is the function of Golgi apparatus? It causes glycosylation of protein. So that is done in this Golgi apparatus proper. So this is the diagram. We have this cis phase, we have this trans phase, and we have this Golgi apparatus proper. Cis phase is also known as the receiving end because it receives the protein. Trans phase is also known as the releasing end because because यहाँ से हमारा glycosylated protein is released from this end. Moving further, what is the function? First and important function of our Golgi apparatus is post-translation modification. What happened? After translation of protein, The modification of protein occurs in our Golgi apparatus. So what happened? Protein synthesis occur in our rough endoplasmic reticulum. They move to the Golgi apparatus through cis phase, and what happened? Carbohydrate side chain is added to the protein to form this glycoprotein, and the function that is done by this Golgi apparatus, we can say the glycosylation of protein is done by this Golgi apparatus proper, and it ensures the stability of protein. Yeah, you can ask any query. If you have any query, you can ask in the session. Post-translation modification is done by our Golgi apparatus proper, and what does it do? It causes glycosylation of protein, and that glycosylation of protein is required for stability of proteins. Next cellular organ is the lysosome. They are the vesicular organ that is separated from our Golgi apparatus. Okay? And they finally disperse throughout the cytoplasm. So what happened? This proton pump is present in our this lysosome. They causes concentration of hydrogen ion inside the lysosome. So that is responsible for acidic pH of lysosome, and this acidic pH of lysosome is going to cause the destruction of substances. Is this clear? So lysosome are vesicular structure that is separated from our Golgi apparatus. That consists of proton pump that is responsible for acidic pH of lysosome, and that acidic pH of lysosome is responsible for destruction of substances. So lysosome, what does it do? It causes degradation of worn out organelle. It causes degradation of excess secretory product. It is going to cause degradation of bacteria. It is going to cause degradation of this aged protein. So it is also known as suicidal bag or the residual body. The enzyme present in our lysosome are acid phosphatase, acid hydrolase, and the acid lipase. So do remember, the enzyme present in our lysosome are acid phosphatase, acid hydrolase, and the acid lipase. Next important function of lysosome is autophagy or self-destruction. This is also done by lysosome. So what happened during the starvation? Our lysosome engulf the mitochondria. So protein inside the inner mitochondria will be metabolized, and that is going to provide the ATP. So what happened? This phenomena is known as autophagy and self-destruction, and that occurred during starvation. So what happened during starvation? Lysosome is going to engulf and destroy mitochondria. Protein from the inner mitochondria will be released, and whenever this protein is metabolized, energy will be released, and that's how we survive this starvation period. Is this clear with everybody? Anybody, any doubt, you can ask in this session only. So moving further, next cellular organelle, that is a peroxisome. This is physically similar to the lysosome. It contains oxidase and the catalase, while lysosome was consisting this acid hydrolase, but this peroxisome is containing this oxidase and catalase. What is the function of lysosome? It breaks down the excess fatty acid, and they are responsible for destruction of free radical like hydrogen peroxide by the catalase enzyme. They are responsible for oxygen utilization, and they are going to accelerate the gluconeogenesis, means new glucose formation is promoted by this peroxisome. Next, coming to the cytoskeletal filament. The cytoskeletal filament is the cellular organelle that is present throughout our cytoplasm, and they provide the strength to the cell, and they determine the shape of cell. So there are three types of major protein component in our cytoskeletal filament: microtubule, intermediate filament, and the microfilament. From microtubule to microfilament, size decreases. So maximum sizes of microtubule and minimum sizes of microfilament. First, coming to this microtubules. So this is the diagram. Our microtubule has maximum diameter. Our microfilament consists of actin filament and a myosin filament. So this actin filament is having the minimum diameter, that is 7 nanometer. Next is the intermediate filament that consists of 10 nanometer diameter. So first, coming to this microtubule. This microtubule is long, hollow in structure. So see, this is the diagram. This is hollow in structure. So this is long, hollow in structure and made up of three proteins: kinesin, dynein, and tubulin. So this microtubule consists of three proteins: kinesin, dynein, and tubulin. So now coming to its function. एक zonal transport आप लोगों ने पढ़ा होगा. It is forward and reverse transport. Anterograde and retrograde transport is there. Anterograde transport में what happen? The transport occur from cell body to synapse, and that occur with the help of this kinesin. Next is the retrograde transport that occur from the synapse to the cell body, and that required this dynein protein. So first role of this microtubule is this axonal transport. Next, this is the diagram. Anterograde transport required this kinesin. Retrograde transport required this dynein. Moving further, dynein also helps of certain organs like fallopian tubule, sperm, and the cilia. What happened? Due to the ciliary movement, sputum is cleared from our nasopharynx, and they are also responsible for rotation of internal organs. So whenever there is absence of this dynein, what happened? The fallopian tubule movement will not be there, sperm movement not will be there, and ciliary movement is also not there. So the most common syndrome out here is having this immotile cilia syndrome, which is also known as a Cardiogenic Syndrome. This Cardiogenic Syndrome consists of a triad of sinusitis, bronchitis, and the situs inversus. What happened? If sinusitis is not treated, it is going to cause this bronchitis. Ciliary movement is also responsible for rotation of internal organs. So if ciliary movement is not there, there will be situs inversus. Along with that, our fallopian tubule, our sperm will not be motile, and immotile sperm and immotile fallopian tube will lead to infertility in both male and female. Infertility in male is also known as asthenospermia, which is also known as immotile sperm. Is this clear? So second role of this microtubule is movement of the cilia, fallopian tubule, and sperm. Moving further, they are also responsible for movement of chromosome during cellular division. They require this tubulin protein. So that's why the microtubule inhibitor is going to inhibit this chromosomal movement, and when chromosomal movement is not there, there will be no cellular division, and that's why it is used in the anti-cancer drug. This microtubule inhibitor are used as a anti-cancer drug. Like our vinca alkaloid, like vincristine, our taxane is like paclitaxel. So this point is important. Do remember, everybody. So all the function of microtubule, is this clear with everybody? Do reply fast. Do reply fast. Shall we proceed further? Okay, moving further to the intermediate filament. Among all cytoskeletal filament, this intermediate filament is the most abundant. The tumor marker for tumor of mesenchymal origin. Glial fibrillary acid protein, they are present in our astrocyte or glial cell, and they are tumor marker for the astrocytomas or gliomas. Lamin is present in the nucleus of cell, and they are the tumor marker for progeria or premature aging. So this chart is very, very, very important. Do remember, everybody. If keratin is present in our epithelial cell, desmine is present in our mast cell, vimentin is present in our connective tissue, glial fibrillary acid protein is present in our astrocyte, lamin is present in our nucleus of cell. Is this clear with everybody? Do reply fast. Moving to the last cytoskeletal filament, that is a microfilament. They are small in size, and two type of microfilament are there: actin and myosin. Globular actin polymerized to form this filamentous actin, and this filamentous actin is responsible for cell motility. Is this clear? So they are also responsible for certain cellular junction. They are also responsible for skeletal muscle contraction. So this is all about cytoskeletal filament. We have microtubule, we have intermediate filament, we have this microfilament. Now coming to the cellular junction. The cellular junction can be intercellular junction or junction that is present between cell to basement membrane. The intercellular junction are zona occludens, zona adherens, desmosome, and the gap junction. In cell to basement membrane, we have this hemidesmosome. We have the cell adhesion molecule like cadherin, integrin, and the selectin. Is this clear with everybody? So, moving to the different type of junction, we have this tight junction, zona occludens, zona adherens. We have this desmosome. We have this gap junction. So in the diagram, what we all are seeing, these four are intercellular junction. They are present between the two cells. In this, the most apical one is tight junction, zona occludens. Okay? And zona adherens is present at the basal of tight junction. Then we have desmosome. Then we have this gap junction. Next is the junction that is present between cell to basement membrane, and in this we have this hemidesmosome and the adhesion molecule like our integrin, cadherin, integrin, and we have this hemidesmosome that is present between cell to basement membrane. So moving further to the detail of each one. First, coming to the zona occludens, which is also known as a tight junction. They are present at the apical margin of cell in the epithelia. They are having this rope-like connection between the cell. Tight junction forming proteins are claudine, occludin, and junctional adhesion molecule. Tight junction forming proteins are claudine, occludin, and junctional adhesion molecule. And this zona occludens, which is also known as the tight junction, they permit passage of some ion and solute between the adjacent cells. So do not confuse कि अगर tight junction है तो that is not allowing any ion or solute passage. They also permit passage of some ion and solute between the two cells. Is this clear? So do remember that consists of certain protein like claudine, occludin and junctional adhesion molecule. So now coming to the location of this tight junction, they are present in our blood-brain barrier, intestine, kidney and the choroid plexus. They all consist of this tight junction zona occludens. Coming to the next one, that is zona adherence. This zona adherence is present at the basal site of tight junction. Diagram में देखा था. Now they connect actin filament of one cell to the actin filament of another cell, and they consist of protein that is known as cadherin. So this is the cadherin, and they connect the actin filament of one cell to the actin filament of another cell. Next is the desmosome, and this desmosome consists of, in this we have the thickening of membrane. This is the thickening of membrane. Both cell has having this thickening of membrane, and the intermediate filament of one cell is connected with the intermediate filament of next cell, and they consist of a protein that is known as desmoglein. Okay? This desmosome is present in area of mechanical stress like in our heart, in our skin and the uterine cervix. So desmosome are present in area of mechanical stress like heart, skin and the uterine cervix. Is this clear? Moving to the next, that is the gap junction. This gap junction permits substance to move from one cell to another cell without moving to the ACF. It has not to go to the VCF. ठीक है? Without going to the ACF, one cell से हमारा दूसरे cell में हमारा ionic movement can occur from this gap junction. The diameter of this gap junction is 0.8 to 1.4 nanometer, and it can permit the passage of iron, sugar and the amino acids. Gap junction forming proteins are connexins, and each connections consists of 6 connexins. ठीक है? हमारा zona occludens, zona adherence, we have this gap junction, we have this desmosome. So zona adherence consists of cadherin, desmosome consists of desmoglein, and this gap junction consists of connexins. This gap junction is a characteristic feature of our heart, neuron and our smooth muscle. Is this clear? Everybody, is this clear? So they are basic intercellular junction. Moving further, this cardiac gap junction are located in intercalated disc. Next is the hemidesmosome, that is present between cell to the basement membrane. As the name suggests, hemidesmosome means look like a half desmosome, and it connects cell to the basal lamina. ठीक है? It connects the cell to the basal lamina. What does it do? It anchors the intermediate filament in one cell to the extracellular matrix. ठीक है? Is this clear? Desmosome क्या कर रहा था? Intermediate filament of one cell को intermediate filament of another cell को connect कर रहा था. Hemidesmosome क्या कर रहा है? Intermediate filament of one cell से extracellular matrix को connect कर रहा है, and it consists of integrate. Desmosome was consisting of desmoglein, and this is consisting this integrate. Okay, now coming to the question part. Everybody ready for the question? First question of today's session. Cell membrane of a cell is freely permeable to 3 ion X, Y, Z. At resting membrane potential, the respective potential by X and Y is -30 and -40 millivolt. What will happen to the membrane potential if a drug is infused to block the permeability of this Z? It is going to cause the depolarization, hyperpolarization, no change, or hyperpolarization followed by repolarization. Everybody answer this question. X is responsible for minus 30, Y is responsible for minus 40. So we all know summation of potential by the all ion is 0. So Z is responsible for minus X and minus Y. So it is responsible for plus 70 millivolt. So it is causing the plus millivolt. So अगर plus नहीं, अगर Z ion हमारा diffuse होता, it is going to cause this plus 70 millivolt. Give me 1 sec. So it is going to cause this plus 70 millivolt. So what happened? If Z ion is not moving, it is going to cause not causing this plus 70 millivolt. So it is going to cause negativity inside the membrane. So it is going to cause this hyperpolarization of membrane. So answer is B. Coming to the next one, X, Y and okay, same question है, skip करते. Coming to the next question, plasma membrane is mainly composed of cholesterol, carbohydrate, phospholipid or the protein. The major component of our cell membrane is the protein. 55% is protein. Coming to the next one, eukaryotic plasma membrane is made up of all except carbohydrate, triglyceride, lecithin or the cholesterol. Eukaryotic plasma membrane is made up of all except. Everybody answer this question. Eukaryotic plasma membrane is made up of all except our plasma membrane consists of carbohydrate, they consist of lecithin, they consist of this cholesterol, they doesn't consist this triglyceride. Coming to the next question, which of the following major mineral is a component of biological membrane? So the major mineral is phosphorus. So coming to the next question, true about lipid bilayer of cell membrane is asymmetrical arrangement of cell membrane component, lateral diffusion of ion, symmetrical arrangement of cell membrane component not made up of amphipathic lipids. True about lipid bilayer of cell membrane. Everybody answer this question. True about lipid bilayer of cell membrane. Lipid layer provide asymmetrical arrangement to our cell membrane. Different type of lipid are there. Some are present at outer side, some are present at both sides, some are present only at the inner side. So they provide asymmetrical arrangement to cell membrane. Coming to next question, in cell membrane following are true. Lipid are regularly arranged, lipid are symmetrical, protein displaced laterally or none. In cell membrane following are true except, in cell membrane all are true, following are true. Everybody answer this question. In cell membrane, protein are displaced laterally. So answer is C. Coming to next question. Membranal fluidity is increased by stearic acid, palmitic acid, cholesterol, or the linoleic acid. Membranal fluidity is increased by Everybody answer this question. Membranal fluidity is increased by linoleic acid, which is a unsaturated fatty acid, essential fatty acids. Coming to the next one, function of cholesterol in plasma membrane is help in transport of iron, help in exocytosis, maintain fluidity, help in synthesis of bile acids. Function of cholesterol is everybody answer this question. Function of cholesterol in the plasma membrane is everybody answer this question. Function of cholesterol and plasma membrane is it maintain the membranal fluidity. So answer is C. Coming to the next one. Decreased protein lipid ratio is seen in inner mitochondrial membrane, outer mitochondrial membrane, sarcoplasmic reticulum, or myelin sheath membrane. Decreased protein lipid ratio is seen in decreased protein lipid ratio is seen in means lipid concentration is more in our myelin sheath membrane. Our nerve cell membrane had more lipid concentration. So answer is D. Coming to the next question, on a weight basis the membrane contain protein and lipid in which ratio? 1:2, 1:1, 2:1, or 4:1? On a weight basis, the membrane contains protein and lipid in which ratio? On a weight basis, the membrane contains protein and lipid in which ratio? They contain each one an equimolar ratio. Coming to the next one. Peripheral cell membrane proteins are pump, channel, adhesion molecule or the enzyme receptor. Peripheral cell membrane proteins are everybody answer this question. Peripheral cell membrane proteins are, yeah, very true, the adhesion molecule. Answer is C. Coming to the next one. Most permeable to pure phospholipid bilayer is oxygen, sodium chloride or water. Most permeable to pure phospholipid bilayer is most permeable to pure phospholipid bilayer is most permeable to phospholipid bilayer is oxygen. All the gases are most permeable to our phospholipid. All the lipid soluble substances are most permeable. So answer is A. Coming to the next one, most diffusible ion across cell membrane is sodium, potassium, chloride or none. Most diffusible ion across the cell membrane is Most diffusible ion is sodium ion. Do you remember? Answer is A. Coming to the next one. A cell membrane is damaged by insertion of a microneedle. Repair shall occur by which of the following process? Lateral movement of protein, resealing of lipid bilayer, enzymatic reaction, hydrophobic interaction. A cell membrane is damaged by insertion of a microneedle. Repair shall occur by which of the following process? Cell membrane is damaged by insertion of microneedle. Repair shall occur by which of the following process? Everybody answer this question. So when we damage the cell membrane by inserting a microneedle, there is no cytoplasmic loss, and when there is no cytoplasmic loss, repair occur by the lateral movements of lipid. Coming to the next question. Which of the following is not a monomeric intermediate filament? Desmine, tubulin, keratin or vimentin? Desmine, tubulin, keratin or vimentin. Which of the following is not an intermediate filament? Excuse me. Which of the following is not an intermediate filament? Tubulin is a microtubule, not an intermediate filament. So answer is B. Very good. Coming to the next one. Transport across nucleus is all by except cabulin, local signal, importin or the rat protein. Transport across nucleus is by all except. Everybody answer this question. Transport across nucleus is by all except. Transport across nucleus is by all except. Importin is helping in the transport across nucleus. So rat protein is not required for the transport across nucleus. So answer is D. Coming to the next question. Energy production enzymes are located in rough endoplasmic reticulum, ribosome, Golgi apparatus or the mitochondria. Energy production enzymes are located in energy production enzymes are located in? Energy production enzymes are located in mitochondria. Very true. Coming to the next question, ribosomes are attached to nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, or the mitochondria. Ribosomes are attached to excuse me, ribosomes are attached to, I guess, easy. They are attached to obviously rough endoplasmic reticulum. Coming to the next question, protein synthesis occurs in smooth endoplasmic reticulum, Golgi body, rough endoplasmic reticulum, or the lysosome. Protein synthesis occurs in everybody answer this question. Protein synthesis occurs in everybody answer this question fast. Protein synthesis occurs in rough endoplasmic reticulum. Answer is C. Very true. Coming to the next one, intracellular sorting and packing is done in endoplasmic reticulum, Golgi apparatus, ribosome, or the nucleus. Chalo, everybody answer this question. Intracellular sorting and packing is done in intracellular sorting and packing is done in intracellular sorting and packing of protein occurs in, yeah, very true, it occurs in our Golgi apparatus. Answer is B. Coming to the next one, catabolism of hydrogen peroxide is carried out by peroxisome, mitochondria, endoplasmic reticulum, or the lysosome. Everybody answer this question. Catabolism of hydrogen peroxide is carried out in Everybody answer this question. Yeah, very true. It occurs in the peroxisome. Coming to the next one. All other marker of mitochondria except ATP synthetase, 5-nucleotidase, glutamate dehydrogenase or creatine kinase. All are the marker enzyme for mitochondria except. Everybody answer this question. All are the marker enzyme for the mitochondria except. Yeah, way true. All are the marker enzyme for mitochondria except our 5-nucleotide. Very good, everyone. This is the summary जो आपको याद रखना है. Mitochondria for mitochondria marker enzyme are monoamine oxidase, ATP synthase, succinate dehydrogenase. For lysosome, the marker enzyme is acid phosphatase. For peroxisome, we have catalase, we have oxidase. For endoplasmic reticulum, we have glucose 6-phosphatase. Excuse me. For Golgi apparatus, we have galactosyl transferase, we have cellular transferase. For the plasma membrane, the marker enzyme is 5-nucleotidase. For cytoplasm, we have the selectate dehydrogenase. So all these marker enzymes are very important from the exam point of view. Do remember, everybody. Coming to the next question, a marker enzyme of Golgi apparatus is galactosyl transferase, acetyl coenzyme A synthetase, pyruvate kinase, or melanin coenzyme A. All are marker for Golgi apparatus except all are marker. A marker for Golgi apparatus is everybody answer this question. A marker of Golgi apparatus is the marker of Golgi apparatus is galactosyl transferase and the cellular transferase. अभी just discuss किया था. Is this clear with everybody? It's not pyruvate kinase or it's not acetyl coenzyme A synthetase. Coming to the next question. Okay, we will take further these questions in another session. Some another session we will take these questions. Okay, we are ending this session. Anybody any doubt you can ask in the session. आज के सेशन से रिलेटेड कोई भी डाउट हो तो क्लियर कर सकते हैं. Hope you all like the session. If you like the session, do like and share. This is important. जितना ज़्यादा share करते हो उतनी class interactive होती है. So do share everybody and subscribe our channel, that is Let's Crack NEET PG, and press the bell icon so that you will get notification of every class and you won't miss a single class. So follow us on the Unacademy page also. My page on Unacademy is unacademy.com/@KGMC. You will get the link of this page on the app description box also. So follow me out there so that you will get to join many more plus classes, special classes. Regular मेहनत करते रहिए, you will definitely able to achieve your target. So let's crack it together. See you all in the next session. This is the code Dr. Nidhi. If you want to join any paid course, you can use this referral code to get additional 10% off. That's all for today. Thank you so much for joining. See you all in the next session. Bye bye everybody.